Thank you for joining the 2025 Neuropixels course. This is a course that we offer every year. My job is just to give you a brief introduction to the course. Most of the information is on this website. So if you Google Neuropixels 2025 course, you'll get there. I am technically one of the organizers, but in practice, all the work was done by Flora and Annie. Here they are, Annie and Flora. They're both postdocs in the lab, and they're very, very, very expert in Neuropixels. I'm just going to give you a whistle-stop tour of history with Neuropixels, but then Tim Harris and others will talk about it in much more detail. This is a graph that shows the number of recording sites per shank of a probe over time, and you can see that Neuropixels made a huge difference. Because suddenly you have in the order of 1,000 recording sites in a single shank. And what is a shank? A shank is a very thin electrode or probe that you put in the brain. It's 70 microns wide, so it doesn't cause any noticeable damage. And along this shank, you have about 1,000 recording sites, depending on which probe. And one of the beautiful things is that what comes out is already fully digital, so it could be in principle attached directly to a laptop. You don't need a lab. In practice, you cannot attach it directly to a laptop because the probe doesn't speak USB or FireWire or whatever codes a laptop would understand, so you need to go through an intermediate box. Now, this 12,000 probes delivered to 700 labs, this was true 3 years ago, so I think at this point, many more probes have been delivered to many more labs. I should update this. When the probe was developed, and this was done by a consortium headed by Tim Harris at Howard Hughes Medical Institute in collaboration with a bunch of institutions, and he will be the one who speaks right after me. It was revolutionary to be able to record from hundreds of neurons at the same time. So here's an example where we inserted 2 probes at the same time in the mouse brain, and the first probe up here goes through a motor cortex and striatum and gave us 397 neurons. The second probe was going through visual cortex, hippocampus, and thalamus, and gave us 344 neurons. This is unprecedented. This was amazing in 2017. Maybe it's normal now. What it became possible also was to do a dense sampling of neural activity across the brain, and this is an example where 30,000 neurons were recorded from 10 mice, sampling a lot of the one hemisphere. Last month, there was a study published by the International Brain Laboratory where this number, instead of being 30,000, is now 600,000. There have been various versions of the probe, and I just want to introduce some of the words that you might hear in the rest of the course. So there is the 1.0 probe, which was published in 2017 and released 2 years later in 2019. And it's illustrated here, and it had about 1,000 recording sites, actually 960. Then there's the 2.0 probe, which was released in 2023, so 2 years ago. It has 1 shank or 4 shanks, depending on how many you want. And it has 5,000 recording sites if you get the 4 shank one. And the arrangement of the sites is slightly different. They're arranged in a line. And as I hope you will hear in other talks, this allows you to track neurons and do correction for the motion of the brain relative to the probe. This also allows you to record from the same neurons for weeks or months with chronic implants. And these are 3 figures from this science paper showing that you can have a lot of neurons for a lot of days, and these neurons tend to be the same neurons as illustrated by this graph, where you have 2 neurons in the visual cortex of a mouse recorded 10 days apart, and their responses to images stay very similar. And of course, stability is probably decreasing with time, but it's still remarkable. You can follow the same neurons for weeks and months. Now, I told you about Neuropixels 1.0 and Neuropixels 2.0. There's more Neuropixels, so to just introduce this, it's starting to be a bit complex. It all started with Neuropixels 1.0, published in 2017. Then there was Neuropixels 2.0, published in 2021. From 1.0, there was a child called Ultra. Oh, this is in press, in Neuron. I should update this. And Ultra is just like 1.0, but the sites are much more densely near each other. And this would allow you to get very small neurons or axons, or in some brain regions, maybe it's the only way to go, like maybe for certain cerebellar cells, this is really what you need. Now, if a probe is in green, you can order it directly from neuropixels.org. If it's in yellow, you need to place a special order, but they might build it for you. They don't have to, but they might. And by the way, neuropixels.org is run by IMEC. IMEC is a non-profit research institute, and probes are sold at cost price. Nobody's making money for these probes. Then there's another version of 1.0 that is longer, and it's called NHP for non-human primate. It has been placed, oh, this is published, I should update this. This is in Nature Neuroscience. And you can buy these probes and put them in large brains like those of non-human primates, but also these probes have been put in the brains of humans, but only during surgery, because you cannot leave them there. You need to put them in and then take them out. And hopefully you will hear about this in the course. Then from 2.0, there is Opto, which I'll show you one slide about, which is a probe that can also emit light for optogenetics. And then there's the next version, which is revolutionary, which is being developed again by Tim Harris and his team. And I don't quite know if the name is NXT or 3.0. And hopefully Tim will tell us in the next talk. And it's in development. So the red ones are in development. You cannot buy one. The green ones are available, and the yellow ones are probably available as a special order. So let me just show you one slide about NeuroPixels Opto, and then I'll tell you a few things about you, not about me or NeuroPixels. Opto is a probe that can also emit light. There's a bioRxiv paper this year out, and hopefully soon in press. And what you can do is, if you insert it in the cortex, you can activate different groups of neurons, as shown in this histogram, with different emission sites. So this is a figure from the paper where you see that there are a bunch of neurons that can be activated by visual stimuli, because this is visual cortex. You can also activate them by putting a laser outside the brain using optogenetics, and through emitter stimulation, which is the job of these NeuroPixels Opto. Now, a couple of slides about who you are. Some of you are undergrads, some of you are master's students, some of you are PhD students, some of you are postdocs, a few principal investigators. If you didn't understand what I talked about up to now, this course is probably too advanced for you. If instead you were comfortable or almost comfortable, this course is probably okay for you. You're half and half male, female. This is the same information. No, this is your level of training. Some of you, your highest level is a BA, some of you a master's, some of you are PhD, and some of you learned on the job. This is where you are physically now in terms of where your institution is. We are delighted to see a large contingent from Egypt. Of course, your origin is much more broad, and there's a much more broader distribution of origin. NeuroPixel experience is beginner by large, but there's also quite a few who are setting up for experiments, but also a lot who have no idea what NeuroPixels is. So don't know how much you're going to get out of this course if you have no idea what NeuroPixels is. And most of you want to do acute recordings. Some of you want to do chronic but recoverable. And you will hear about what this means and the implications in the rest of this amazing course. And I yield the floor to the next speaker. Thank you so much.